The structure of the genome. DNA is a macromolecule and an assemblage of a large number of atoms bound together in a defined arrangement whose three-dimensional structure can be described by techniques such as X-ray crystallography. But DNA is also an information repository, which is a property more difficult to describe in simple molecular terms. If, as noted above, a gene corresponds to a particular segment of DNA, then the sum of the genetic information that an individual organism inherits from its parents is equivalent to the sum of all the DNA segments present in the fertilized egg at the start of life. All the individuals that make up a species population share the same set of genes, even though different individuals invariably possess slightly different versions or alleles of many of these genes. Thus, each species of organism has a unique content of genetic information which is known as its genome. For humans, the genome is essentially equivalent to all of the genetic information that is present in a single haploid set of human chromosome, which include 22 different autosomes and both the X and Y sex chromosomes. The complexity of the genome. To understand how the complexity of the genome is determined, it is first necessary to consider one of the most important properties of DNA double helix, its ability to separate into its two component strands, a property termed denaturation. DNA denaturation, as was suggested by Watson and Crick, and the two strands of DNA molecule are held together by weak non-covalent bonds. When DNA is dissolved in saline solution and the solution is slowly warmed, a temperature is reached when strand separation begins. Within a few degrees, the process is generally complete and solution contains single-stranded molecules that are completely separated from their original partners. The progress of thermal denaturation or DNA melting is usually mentioned by following the increase in absorbance of ultraviolet light by the dissolved DNA. Once the two DNA strands are separated, the hydrophobic interaction that result from base stacking are greatly decreased, which changes the electronic nature of the bases and increases their absorbance of ultraviolet radiation. The rise in absorbance that accompanies DNA denaturation is very important. The temperature at which this shift in absorbance is half completed is termed the melting temperature or TM. The higher the GC content, %D plus %C of the DNA, the higher the TM. This increased stability of GC containing DNA reflects the presence of extra hydrogen bond between the bases as compared with AT pairs. Then DNA denaturation. The separation of the two strands of the DNA duplex by heat is not an unexpected finding, but the reassociation of single strands into a stable double-strand molecule with correct base pairs seems almost inconceivable. However, in 1960, Julius Marmor and Paul Doty at Harvard University found that if they slowly cooled a solution of bacterial DNA that had been thermally denatured, the DNA regained the properties of the double helix. It is absorbed less ultraviolet light and once again behaved like a genetic material, is being able to transform bacterial cells. It becomes apparent that these studies that supplementary single-stranded DNA molecules were capable of reassociating an event termed renaturation or reannealing. This finding has proved to be one of the most valuable observations ever made in molecular biology. On one hand, reannealing has served as a basis for an investigation into the complexity of the genome, subject discussed in the following section. On the other hand, reannealing had led to the development of methodology called nucleic acid hybridization, in which complementary strands of nucleic acid from different sources can be mixed to form double-stranded hybrid molecules. Examples of the types of questions that have been answered by allowing single-stranded nucleic acids to hybridize are very important, and Nucleic acid hybridization plays a key role in our most important modern biotechnology, including DNA sequencing, DNA cloning, and DNA amplification. Next topic, the complexity of viral and bacterial genomes. Several factors determine the rate of renaturation of a given preparation of DNA. They are the ionic strength of the solution, the incubation temperature, the concentration of DNA, the period of incubation, and the size of the interacting molecule. With these factors in mind, consider what might happen if one were to compare the rate of renaturation of three different DNAs, each constituting the entire genome. A small virus such as MS2, a larger virus such as T4 nucleotide pairs, and a bacterial cell such as E. coli. The primary difference between these DNA is their length. To compare their renaturation, it is important that the reacting molecule are of equivalent length, typically about 1000 base pairs. DNA molecules can be fragmented into pieces of this length in various ways, one of which is to force them through a tiny orifice at high pressure. When these three types of DNA all present at the same length and DNA concentration are allowed to reanneal, they do so at distinctly different rates. The smaller the genome, the faster the renaturation. The reason becomes apparent that one considers the concentration of complementary sequences in the three preparations. Because all three preparations have the same amount of DNA in a given volume of solution, it follows that the smaller the genome size, the greater the number of genomes present in a given weight of DNA, greater the chances of collision between complementary fragments. Next topic, the complexity of eukaryotic genomes. The renaturation of viral and bacterial DNA occurs along single symmetrical curves. Renaturation occurs along such curves because all of the sequences are present, with the exception of a very few species in bacterial DNA at the same concentration. Consequently, every nucleotide sequence in the population is likely to find a partner in a given time at any other sequence. This is a result that would have been predicted from the various gene mapping studies that suggested that DNA of a chromosome contained one gene after another in a linear array. The denaturation kinetics of viral and bacterial genomes contrast sharply with those obtained on mammalian DNA by Roy Britton and David Coheney of Carnegie Institute of, I'm not sure about the pronunciation, Washington, DC, unlike those of the simpler genomes. The DNA fragments from a mammalian genome reanneal at markedly different rates. These differences reflect the fact that the various nucleotide sequences in a preparation of eukaryotic DNA fragments are present at markedly different concentrations. This was the first insight that eukaryotic DNA is not just a simple progression of one gene after another, as in a bacterium or virus, but has a much more complex organization. When DNA fragments from plants and animals are allowed to reanneal, the curve typically shows three more or less distinct steps, which correspond to the reannealing of three broad classes of DNA sequences. The three classes reanneal at different rates because they differ as to the number of times their nucleotide sequences repeated within the population of fragments. The three classes are termed the highly repeated fraction, the moderately repeated fraction, and non-repeated fraction. Highly repeated DNA sequences and highly repeated fraction constitute somewhere from about 1 to 10% of the total DNA. Highly repeated sequences are typically short, a few hundred nucleotides at their longest, and present in clusters in which a given sequence repeats itself over and over again without interruption. A sequence arranged in this end-to-end manner is said to be present in tandem. Highly repeated sequences fall into several overlapping categories, including satellite DNA, monosatellite DNA, and microsatellite DNA. Satellite DNAs consist of short sequence about 5 to few hundred base pairs in length. that form very large clusters, each containing up to several million base pairs of DNA. In many species, the base composition of these DNA segments is sufficiently different from the bulk of the DNA that fragments containing the sequence can be separated into a distinct satellite band during density gradient centrifugation, hence the name satellite DNA. Satellite DNA tend to evolve very quickly, causing the sequences of these genomic elements to vary even between closely related species. The localization of satellite DNA within the centromeres of chromosomes is described later. So, satellite DNA consists of short sequences about 5 to few hundred base pairs in length that form very large clusters, each containing up to several million base pairs of DNA. In many species, the base composition of these DNA segments is sufficiently different from the bulk of the DNA that fragments containing the sequence can be separated into a distinct satellite band during density gradient centrifugation. That's why it is named like that. Satellite DNA tends to evolve very rapidly, causing the sequence of these genomic elements to vary even between closely related species. The localization of satellite DNA within the centromeres of chromosome is described later. Then mini satellite DNA. Mini satellite sequences range from about 1 to 100 base pairs in length and are found in sizable clusters containing as many as 3,000 repeats. Thus, mini satellite sequences occupy considerably shorter stretches of the genome than do satellite sequences. Mini satellites tend to be unstable, and the number of copies of a particular sequence often increases or decreases from one generation to the next, most likely as the result of unequal crossing over. Consequently, the length of a particular mini satellite locus is highly variable in the population, even among members of the same family, because they are so variable or polymorphic in length. Many satellite sequences form the basis for the technique of DNA fingerprinting, which is used to identify individuals in criminal or paternity cases. Microsatellite DNA. Microsatellites are the shortest sequences. Microsatellites are the shortest sequences, 1 to 9 base pairs long, and are typically present in small clusters of about 10 to 40 base pairs in length, which are scattered quite evenly through the genome. DNA replicating enzymes have trouble copying regions of segments that contain these small repetitive sequences, which cause the stretches of DNA to change in length through the generations. Because of their variable lengths within the population, microsatellite DNA have been used to analyze the relationship between different human populations. As illustrated in the following example, it is generally agreed that modern humans arose in Africa. If this is true, then members of different African populations would exhibit greater DNA sequence variation than human populations living on other continents, because genomes of African populations have had longer to diverge. The argument of African genesis has received support from numerous studies on human DNA sequences. In one study that analyzed 60 different microsatellite loci, it was found that members of African population had a significantly greater genetic divergence than Asian or European populations. The vast majority of microsatellite loci occur outside of genes, and the changes in their length generally go unnoticed. This is not the case for the microsatellite sequences discussed in the accompanying human perspective later. So, in DNA fingerprinting, we mentioned earlier about that technique. So, in this technique, which is used widely to identify an individual from a sample of DNA, the DNA is digested by treatment with specific nucleases called restriction endonuclease, and the DNA fragments are separated on the basis of length by gel electrophoresis. The location in the gel of DNA fragments containing specific DNA sequences is determined using labeled props with sequence complementary to those being sought. The DNA fragments that bind these probes have variable lengths from one person to the next because of the presence of variable numbers of short tandem repeats or STRs in the genome. Forensic labs typically analyze about 13 STR marks that are known to be highly polymorphic. The chance that two individuals will have identical STR profiles is astronomically small. The fingerprint shown in this figure, which is not shown, is a criminal case in which the defendant was charged with the fatal stabbing of a young woman. The bloodstains in the pants and shirt of the defendant were compared to the known blood stains of the victim and defendant. DNA from the bloodstains of the defendant clothing did not match his own blood standard, but they match with that of the victim. The lanes contain DNA from the following sources: 1, 2, 3, 4 are control of DNA samples that serves quality control checks. Then defendant blood stain from the defendant's pants, and blood stain from the defendant's shirt, victim's blood. And with the advent of DNA amplification techniques like PCR, minute samples of DNA can be used for this analysis. Once it becomes apparent that eukaryotic genome contains large number of copies of short DNA sequences, researchers sought to learn where these DNA sequences are located in the chromosome. Are satellite DNA sequences, for example, clustered in particular parts of a chromosome or scattered uniformly from one end to the other? It was noted above that the discovery of DNA annealing led to the development of a vast nucleic acid hybridization methodology. The ability to locate satellite DNA sequences illustrates the active analytic power of nucleic acid hybridization. In the re-annealing experiments, complementary DNA strands were allowed to bind to one another while colliding randomly in solution. An alternate experiment protocol called in situ hybridization was developed in 1969 by Mary Lou Part and Joseph Call of the Yale University and was used to determine the location of satellite DNA. The term in situ means in place, which refers to the fact that the DNA of the chromosome is kept in place while it is allowed to react with a particular preparation of labeled DNA. In early in situ hybridization studies, the DNA to be detected or the probe DNA was radioactively labeled and was localized by autoradiography. The resolution of the technique has been increased using probes that are labeled with fluorescent dyes and then localized with a fluorescence microscope. The later technique is called fluorescence in situ hybridization or FISH and has become so refined that it can be used to map the locations of different sequences along single DNA fibers. To carry out nucleic acid hybridization, both interactants must be single-stranded. In the experiment, the chromosomes from a mitotic cell are spread on a slide and the DNA is made, for example, it's an exceptional case, made single-stranded by treating the chromosome with a hot salt solution that causes the DNA strands to separate and remain apart. During the ensuing hybridization step, the denatured chromosomes are incubated with the solution of biotin labeled single-stranded satellite DNA, which binds selectively to complementary strands of immobilized satellite DNA located in the chromosome. Following the incubation period, the soluble unhybridized satellite DNA is washed away or digested, and the sites of the bound labeled DNA fragments are revealed. Satellite DNA is located in the centromeric regions of the chromosome. So when we conduct fluorescence in situ hybridization and the localizing of satellite DNA, the steps that are taken in carrying out fluorescence in situ hybridization in this technique, certain of the nucleotides in the probe DNA are linked covalently to a small organic molecule, usually biotin. Following hybridization, the location of the bound biotin labeled DNA can be visualized by treating the preparation with fluorescently labeled avidin, a protein that binds to biotin with very high affinity. Chromosomes in these preparations usually appear red because they have been counterstained with propydium iodide. Localization of alpha satellite DNA at the centromere of human chromosome. The location of the bound biotin labeled satellite DNA is revealed by yellow fluorescence, which stands out against the background of red counterstained chromosomes. Fluorescence appears only at the site where each chromosome is constricted, which marks the location of the centromere. So mitotic chromosomes are there, glass slide, double-stranded DNA will be there. Treat slide with hot salt solution to denature DNA. Later, single-stranded DNA will be there. Incubate with biotinylated DNA probe, then wash to remove unhybridized DNA. In this example, satellite DNA is employed. Then double-stranded DNA hybrid is formed. Later, incubate with fluorescently labeled iodine to reveal location of bound labeled DNA probe. Counterstain chromosomes, so they appear red. Localization of satellite DNA in centromeres. Next topic, moderately repeated RNA DNA sequences. The moderately repeated fraction of the genomes of plants and animals can vary from about 20 to more than 80% of the total DNA depending on the organism. This fraction include sequences that are repeated within the genome anywhere from a few times to 10 of thousands of times. Included in the moderately repeated DNA fraction are sequences that code for non-gene products, either RNAs, proteins, and those that lack a coding function. Then repeated DNA sequences with coding functions. These fractions of the DNA include the genes that code for ribosomal RNA as well as those that code for an important group of chromosomal proteins, the histones. The repeated sequences that code for each of these products are typically identical to one another and located in tandem array. It is essential that the genes encoding ribosomal RNAs present in multiple numbers because these RNAs are required in large amounts and their synthesis does not benefit from the extra amplification step that occurs for protein coding genes, in which each mRNA acts as a template for the repeated synthesis of a polypeptide. Even though the production of histones does involve an immediate intermediary messenger RNA, so many copies of these proteins are required during early development that several hundred DNA templates must be present. Then repeated RNAs that lack coding functions. The bulk of the moderately repeated DNA fraction does not encode any type of product. Rather than occurring as clusters of tandem sequences, the members of these families are scattered or interspersed throughout the genome as individual elements. Most of those repeated sequences can be grouped into two classes that are referred to as SINEs, or short interspersed elements, and lines, like long interspersed elements. Sign and line sequences are discussed later. Then, non-repeated DNA sequences are initially predicted by Mendel. Classical studies on the inheritance pattern of visible trait led geneticists to conclude that each gene was present in one copy per single haploid set of chromosomes. When denatured eukaryotic DNA is allowed to reannul, a significant fraction of the fragment are very slow to find partners, so slow in fact that they are presumed to be present in a single copy per genome. This fraction comprises the non-repeated single copy DNA sequences, which include the genes which inhibit Mendelian patterns of inheritance. Because they are present in a single copy in the genome, non-repeated sequence localized to a particular site on a particular chromosome. Considering the variety of different sequences present, the non-repeated fraction contains by far the greatest amount of genetic information. Included within the non-repeated fraction are the DNA sequences that code for virtually all proteins other than histones. Even though these sequences are not present in multiple copies, genes that code for polypeptide are usually members of a family of related genes. This is true for the globins, actins, myosin, collagens, tubulins, integrins, and most other proteins in eukaryotic cell. Each member of a multigene family is encoded by a different but related sequence. We will look into the origin of these multigene families in the following section. Now that the human genome has been sequenced and analyzed, we finally have a relatively accurate measure of the DNA sequences that are responsible for encoding the amino acid sequence of our proteins, and it is remarkably small. If you would have suggested to geneticists in 1960 that less than 1.5% of human genome encode the amino acids of our proteins, he or she would have considered the suggestion to be ridiculous. Yet, that is the reality that has emerged from the study of genome sequencing. In the following section, I will try to better understand how the remaining 98% of the DNA sequence might be evolved. But before moving on to that topic, let's learn the human perspective relates to this. Diseases that result from expansion of trinucleotide repeats. For decades, biologists believed that genes were always transmitted from generation to generation as stable entities. On rare occasion, a change occurs in nucleotide sequence of a gene in a germline, creating a mutation that is subsequently inherited. This is one of the basic tenets of Mendelian genetics. Then in 1991, several laboratories reported a new type of dynamic mutation in which the nucleotide sequence of a particular gene changed dramatically between parents and offspring. In each case, these mutations affected genes that contained a repeated trinucleotide unit like CCG or CAG as part of their sequence. In most members of the population, this particular genes contain a relatively small but variable number of repeated trinucleotides, and they are transmitted from one generation to the next without a change in the number of repeats. In contrast, a small fraction of the population possesses a mutant version of the gene that contains a large number of these repeating units. Unlike the normal version, the mutant alleles are highly unstable, and the numbers of repeated units tend to increase as the gene passes from parents to offspring. When the number of repeats increases beyond a critical number, the individual inheriting the mutant allele develops a serious disease. At the present time, more than 20 inherited disorders have been attributed to expansion of trinucleotide repeats. These diseases fall into two basic categories, which we'll consider in turn. Type 1 diseases are all neurodegenerative disorders that result from the expansion of a number of repeats of a CAG trinucleotide within the coding portion of the mutant gene. We can illustrate the nature of the disorders by considering the most prevalent and widely studied in the group, Huntington's disease. Huntington's disease is a familial illness characterized by involuntary uncoordinated movements, change in personality, including depression and irritability, and gradual intellectual decline. Symptoms usually begin in the third to fifth decade of life and increase in severity until death. The normal HD gene, which is stably transmitted, contains between 6 and 35 copies of CAG trinucleotide. The protein encoded by the HD gene is called Huntington, and its precise function remains unclear. CAG is a triplet that encodes the amino acid glutamine. Thus, the normal Huntington polypeptide contains a stretch of 6 to 35 glutamine residue, a polyglutamine tract, as part of the primary structure. We think of polypeptide as having a highly defined primary structure, and most of them do, but Huntington is normally polymorphic with respect to the length of its polyglutamine tract. The protein appears to function normally as long as the length remains below approximately 35 glutamine residues, but if this number is exceeded, the protein takes on new properties and the person is predisposed to developing HD. HD exhibits a number of unusual characteristics. Unlike most inherited diseases, HD is dominant genetic disorder, which means that a person with the mutant allele will develop the disease regardless of whether or not he or she has a normal HD allele. In fact, persons who are homozygous for the HD allele are not more seriously affected than heterozygotes. This observation indicates that the mutant Huntington polypeptide causes the disease not because it fails to carry out a particular function, but because it acquires some type of toxic property, which is referred to as a gain of function mutation. This interpretation is supported by studies with mice. Mice that are engineered to carry the mutant human HD allele in addition to their own normal allele develop a neurodegenerative disease resembling that found in humans. The presence of one abnormal allele is sufficient to cause disease. Another unusual characteristic of HD and other CAG disorders in a is a phenomenon called genetic anticipation, which means that as the disease is passed from generation to generation, its severity increases and or it strikes at an increasingly earlier age. This was once a puzzle feature of HD, but is not readily explained by the fact that the number of CAG repeats in a mutant allele and the resulting consequences often increases dramatically from one generation to the next. The molecular basis of HD remains unclear, but there is no shortage of theories as to why an expanded glutamate tract may be toxic to brain cells. One feature appears indisputable. When the polyglutamine tract of Huntington exceeds about 35 residues, the protein, a fragment cleaved from it, undergoes abnormal folding to produce a misfolded molecule and binds to another mutant Huntington molecule to form insoluble aggregates, not unlike those seen in the brains of Alzheimer's victims, and binds to a number of unrelated proteins that not interact with normal wild type Huntington molecules. Among the proteins found by mutant Huntington are several transcription factors, which are proteins involved in the regulation of gene expression. Several of the most important transcription factors present in cells, including TBP and CBP, contain polyglutamine stretches themselves, which makes them particularly susceptible to aggregation by protein with mutant expanded polyglutamine tracts. In fact, the protein aggregates present in degenerative neurons of HD patients contain both of these transcription factors. The results suggest that mutant Huntington sequences transcription factors disrupting the transcription of genes that are required for the health and survival of affected neurons. This hypothesis was received support from a study in which mice was genetically engineered so that their brain cells lacked the ability to produce certain key transcription factors. These mice exhibited the same type of neurodegeneration as seen in animals carrying a mutant HD gene. The basic neuronal processes, including axonal transport, mitochondrial permeability and fission, cholesterol synthesis, and protein degradation, are also disrupted by the HD mutation and represent possible causes of nerve cell death. It should be noted that a number of HD researchers hold an alternative opinion about the cause of cell death. They argue that it is not the protein aggregates that are toxic, but the soluble mutant protein or fragments of the mutant protein itself. In fact, proponents of this view argue that the mutant protein aggregates protect the cell by sequestering the harmful molecule. It is important for practical reasons to distinguish between these possibilities because a number of proposed therapies are aimed at blocking formation of the aggregates which could actually prove more harmful to a patient. Type 2 trinucleotide repeat disease differ from the type 1 diseases in a number of ways. Type 2 disease arises from the expansion of a variety of trinucleotides, not only CAG. The trinucleotides involved are present in a part of the gene that does not encode amino acids. The trinucleotides are subject to massive expansion into thousands of repeats, and the disease affects numerous parts of the body, not only the brain. The best studied type 2 disease is Fragile X syndrome, so named because the mutant X chromosome is especially susceptible to damage. Fragile X syndrome is characterized by mental retardation as well as a number of physical abnormalities. The disease is caused by a dynamic mutation in a gene called fMRI that encodes an RNA binding protein that regulates the translation of certain mRNAs involved in neuronal development and or synaptic function. A normal allele of this gene contains anywhere from about 5 to 55 copies of a specific trinucleotide CGG that is repeated in a part of gene that corresponds to the 5' non-coding portion of the messenger RNA. However, once the number of copies rose above about 60, the locus becomes very susceptible and the copy number tend to increase rapidly into the 1000. Females with fMRI gene containing 60 to 2200 copies of triplets generally exhibit a normal phenotype, but the carriers of the transmission but are carriers for the transmission of highly unstable chromosome to their offspring. If the repeat number in the offspring race is above 200, the individual is almost always mentally retarded. Unlike an abnormal HD gene, which causes diseases as a result of a gain of function, an abnormal fMRI allele causes diseases as the result of a loss of function. fMRI allele containing an expanded CGG number are selectively inactivated so that the gene is not transcribed or translated. Although there is no effective treatment for any of the disease caused by the trinucleotide expansion, the risk of the transmitting or possessing a mutant allele can be assessed through genetic screening.